Hi, my name is Mark, and I'm a product development manager at TopoGEN. And today I'm going to tell you about our new ICE assay. It stands for the in vivo complex of enzyme. Now, this is a method for detecting covalent topoisomerase DNA complexes in the context of the live cell. And I hope to show you how this kit works and the value of knowing whether a drug hits the target in vivo, that is inside the cell, or not. In order to understand why topoisomerase is such a great target for anti-cancer therapeutics, is you have to understand the reaction mechanism. Topoisomerase enzymes break and reseal DNA, either one strand or both strands, depending on the topoisomerase involved. The breaks are always resealed, and this protects the genome from random DNA damage, which would be detrimental to the viability of the cell, of course. Finally, drugs that disrupt the cycle are very effective anti-cancer agents. And to illustrate that point, I've got a simple animation here. So imagine this is double-stranded DNA. And topoisomerase, what it does is it introduces either a single strand or double strand break. Now here I'm showing just a single strand nick because that's easiest to, it's the easiest to understand. And what happens is the double strand break is generally resealed after breakage. So if you look at the termination of the reaction, you'll see that the break is resealed. So the actual reaction mechanism looks something like this. Now this creates a swivel point where the DNA can untorque, and this is as a result of reading the template. Transcription, DNA replication all requires topoisomerase to release the topological strain on the DNA molecule. The point here is that when there is a single strand break, in this case for topoisomerase 1 as shown here, any drug that comes in and enters into this complex and disrupts the ability of the topoisomerase to reseal will be a potentially excellent anti-cancer drug. So the break then persists in the presence of this drug, and this then leads to cancer cell death from DNA damage. The most important thing from a developmental standpoint in terms of drug development is that this intermediate shown here, which I'm going to use my pointer to outline, this, we call this the covalent topoisomerase DNA intermediate, is important to identify if you're going to find new anti-topo drugs. And this is what the ICE assay does. Again, ICE stands for in vivo complex of enzyme. And it's very important for successful development of drugs because you must be able to identify the complex. So this is basically a test for drug action in the context of live cells, and this is important for a couple of reasons. Many drugs these days are developed using purified enzymes. This is an in vitro process. Such drugs may work in vitro, but they may not enter the cell. In other words, they fail to function in the context of the cell. In addition, testing in a cell context is the most physiological relevant readout that can be done, and so this is important to accomplish this task. So the ICE assay, as I'll show you, is simple and easy, and we have a kit that will provide the technology for you, including antibodies, as you'll see. So here's basically how it works. Imagine you have a cell sitting here, and in the nucleus of the cell, there's DNA in a chromatin milieu, and the topoisomerase is breaking and resealing the backbone of the DNA shown here. And this is what this equilibrium is meant to show. Now the covalent intermediate is this cleavage complex shown here with the red triangle. But the topoisomerase also travels over the DNA in a scanning non-covalent mode looking for binding sites. What happens is we capture this as a freeze frame by adding back a detergent to lyse the cells in the presence of a test drug. And what happens is you arrest the activity of the topoisomerase, and so now your task is to identify this species. So the ICE bioassay will quantify the yield of the top one DNA covalent complex, while at the same time ignoring either free DNA or DNA that's not covalently attached to proteins. So this is the basis of the assay. So the goal is to detect and measure these so-called damage complexes, or as we call them, covalent complexes. But why is it important? Well, this will help validate new drugs. If you identify an agent, a small molecule, or what have you, it would be nice to be able to put it in the context of a cell and see if that drug works as predicted. You can measure the specificity of drugs. For some companies are developing antibiotics, for example, and then they want to test the question, do these antibiotics actually target human cells or human topoisomerase action? It's also a useful biomarker for clinical trials, and in the past we've been involved in some of these studies where you actually can measure covalent complexes in the context of the tumor or in the patient even. So let's go through the procedure. This is the kit we provide. It takes one large petri dish of cells per ICE assay. In other words, if you're testing one particular drug, you would at least need one of these plates. There's roughly 1 million cells, 1 million to 5 million cells in that range. And then you have to include controls. Of course, you'd have to have a positive and a negative control depending on whether you're testing topo-1 or topo-2, and a negative control means there's no drug present. So what you do is you would take one petri dish per assay and treat for about 30 to 60 minutes with the drug. And we try to keep these drug exposures very short because we don't want to kill the cells. So the cells will tolerate short exposure to these toxic agents, and then we come back and we want to catch a snapshot of the activity. So we do a detergent lysis, and we provide reagents in the kit to allow you to purify genomic DNA without the use of proteases. And it's important, obviously it's important to not degrade proteins in purifying DNA because you don't want to destroy the very thing you're trying to measure. And finally, you do a Western blot of genomic DNA, and you compare signals on a per genome basis. And then you can probe with an antibody so it's specific for the particular topoisomerase in question. And this is the kind of result you might see. With no drug, you would have on your slot blot, you have low, medium, and high DNA inputs, you know, precise DNA concentration. In the presence of VP16, this is a top 2 control drug, you get no signal. And in the presence of camptothecin, you get a strong signal here that demonstrates the specificity because camptothecin is specific for a type 1 mechanism. So in this particular experiment, the cells would have been treated over here with camptothecin for 30 to 60 minutes and then processed for topo-1 signals using an anti-topo-1 antibody. So the ICE is simple, quantitative, and it's fast. You can do it in a day. It is important to note that ICE assays can be done in virtually any cell line, so the investigator can choose the cell line to do the experiment. It gives you good physiological relevance because it's done in the context of the cell. It will work in mouse and rat because our antibodies cross over to rodent. And you can compare antibody signals on a DNA basis. This is, in effect, genome equivalence. So, in other words, your readout is normalized on a per cell basis. And you achieve that by putting identical quantities of genomic DNA on the slot blot. There's no need to run costly, labor-intensive cesium chloride gradients. ICE assays are much faster and more convenient. And we have different kits, and they're defined based on the antibody probe. So, for example, if you're looking at top 2 alpha, we have an antibody that's specific for the alpha form, isoform. We can look at top 2 beta. We have top 1. We have top 3 alpha, and we have top 3 beta. So those are the kit selections based on antibody. And all kits include the probe, the antibody, control drugs, very clear protocols, reagents for DNA purification, and technical support. So here's some results you might see. This is an actual experiment where we took exponentially growing cells. These were HeLa cells, and we had a negative control with no drug. We treated with the etoposide, VP16, which is a top 2 drug, positive control, and we treated another dish with camptothecin as a top 1 control. And here are the results. Now these are 2 different blots, and here we probed with a topoisomerase 1 antibody, and here we probed with a topo-2 antibody. And you can see that if you probe this particular blot with topo-1, you can see that we get camptothecin giving a strong signal, which is consistent because camptothecin is a top-1 poison. And the flip side of that is the top-2 alpha probe, where we probed with this particular monospecific antibody, and you can see it only comes up with VP16, which is a top-2 poison. And here you see the approximate amounts of DNA we loaded on this blot. So this is easily recoverable from a single 100 millimeter dish, this amount of DNA. And you can see that less than 1 microgram of DNA is sufficient to get a signal. The TopoGEN ICE assays are now available. Please go to our website, review catalog number 1020 for details and ordering information. We offer excellent technical support for all of our products, and if you have questions, contact us at support@topogen.com. Thanks for your time.